video we are going to discuss about one of the important and advanced technique in molecular biology, which is 2D gel electrophoresis. So from the name itself, we can identify their function. So electrophoresis means separating molecules with electric current. So gel electrophoresis means we are separating these molecules in a gel, and 2D means two dimension. So we are separating the molecules based on their two dimensions, two properties in simple terms. So now what is this 2D gel electrophoresis? A technique used to separate proteins. So in this technique we separate proteins based on their charge and mass. So two dimensions based on their two properties we separate this protein. What are they? They are charge and mass. Now who discovered this technique? Oh, Farrell and Close in 1975. I don't know who is the guy who is standing in the middle, but these two guys separated, discovered this technique in 1975. Now coming to why this technique has been found. Before this, we to separate protein we used the SDS page. But what is the difficulty in that SDS page? It can be separated only based on one dimension, which is based on their size. It cannot separate based on their charge. But there are some proteins with the same size but different charge. There are different proteins, so it cannot distinguish the proteins difference based on their charge. So to differentiate proteins based on their charge, also we use this 2D gel electrophoresis. Now how? So the first dimension is isoelectric focusing. You might think before I said charge, now it is mentioned as isoelectric focusing. Isoelectric focusing is nothing but the charge. We will discuss why it is named like that. Second is SDS page. So same, based on their size. Now let's check how 2D gel electrophoresis is done. Now coming to the first dimension, which is isoelectric focusing. So separating proteins based on their charge. So in that separate in that most particular in that more particularly, we separate molecules based on their isoelectric point. So what is this isoelectric point? It is a particular pH at which the molecules carries no net charge and it will become neutral. So I said before, like there are proteins with positively charged proteins, negatively charged proteins based on their amino acid sequence. So at one particular point, these positively charged amino acids protein will become neutral, or this negatively amino proteins will become neutral. So at that particular point is called as isoelectric point. So for example, if there is a pH scale from 1 to 14, my charges, my isoelectric focusing point is 3. Let's guess 3. So till 1 to 3 I will be positively charged. When reaching the point 3 I will become neutral. If I cross 3, and if I stand in 7 or 8, I will be negatively charged. So to become neutral I have to come to 3 again. So this is the isoelectric point. So this, if the pH is lesser than the pi, the molecule will be positively charged. If the pH is more or higher than the pi, pi is nothing but the isoelectric point, the molecule will be negatively charged. So this is the principle. So protein moves towards the opposite electrodes, and when the pH and pA is equal, then the net charge will be 0 and it will become, the protein will be neutral. So it cannot migrate towards further and it will stop. So each molecule will have different isoelectric point. So some might have isoelectric point as 3, some might have isoelectric point as 5, and some might have 2 or 1. So based on that, it will be we will change the pH and it will be getting separated at their different points. So this is the first dimension, separating the protein based on their charge. Now coming to the procedure of the first dimension, we use IPG strip, which is immobilized pH gradient strips. So in this only we will be loading our sample and it will be separating based on their charge. So what this IPG strip contains is acrylamide and various variable pH ampholites. So with that only we will vary our pH. Apart from this, we will add dehydration solution also. In this solution we will be containing urea, DTT and etc. So why we add this dehydration solution is to denature protein structure. Some proteins will be in 3D structure and complicated structure, so it will be very difficult to move. So for that we denature their structure, and then we will be applying the electric current. So high voltage of 10,000 volt will be applied in this IPG strip, so that our protein molecules will be separated, and then it takes a long period of time. So around 10 hours it takes to separate this protein molecules based on their size in this IPG strip. So we will be placing in the IEF apparatus, so isoelectric focusing apparatus, to performing this procedure. So coming to the second dimension, which is size, which is nothing but the same SDS page. So we will we separated the protein molecule based on their charge. Now we take that IPG strip and place it in the SDS page well. So in the SDS page we will be containing sodium dodecyl sulfate. So SDS page, all the proteins will be negatively charged and it will be start moving, separating based on their size. So smaller molecules will move faster and reach the bottom of the gel, and it will be get separated. But the larger molecule will be moving slower. Thus, the protein will be separated based on their size. So this is the second dimension. So after completion of this 2D gel electrophoresis, this is how our result will be looking. So you can see that blue color dots are the protein molecules which are separated based on their size and charge. So in bottom you can see, in x-axis you can see from 3 to 10 it is the IPG strip pH gradient. So for example, if I want to locate what is the charge and size of the protein molecule, which is 5. So the 5 is located near 6. So that is the charge of the molecule. And coming to the size, it's in the y-axis you can see from 15 to 100. So near around molecule 5 is located near equal to the 40. So its size might be 40 kb or something. So with this we can separate the protein molecule based on their size and charge, and we can identify their properties. Coming to the application, it is used in molecular biology studies, protein qualitative studies, peptide mapping, like protein quantification also is done. So coming to the advantages, better resolution of the sample is obtained. So based on the charge and mass, we like in SDS page only one dimension is known, but in 2D gel electrophoresis we get a better resolution of the protein, and more number of samples can be analyzed at the same time. So what are the limitations? Mean less abundant protein cannot be analyzed. So and it's a very time consuming process. So for the first dimension it take around 10 hours. So it is a very time consuming and labor intensive process. It's not easy like we turn on a switch and it will start running. Everything has to be done manually, and it should be done very carefully. So even a small mistake might not give the proper result. So these are the limitations of this 2D gel electrophoresis. At last, in this video, we check the important details of 2D gel electrophoresis, its principle, its important materials, its procedure, how it is done, and how the results are interpreted, application, advantage, disadvantage, and every important things. So that's all.